modify angle, this light, so you can see it's reflecting.
You can see the light over there. It's reflecting.
Right? And then, so if you see a kind of, I mean,
You see a tiny bit of light appear right there.
yeah. So if you see like a yellow light in the bottom layer, that means it is in its that critical angle. So now it cannot go beyond that, something like that. So we can adjust the light to make.
So, Present, 1 second. So now you see that very little light appearing at the interface, right? Very small amount of light. So that is very critical for the microscopy technique we use, because you know when you have a cell like this, when you have a fluorescence molecule in the cell, it will have everywhere on the membrane inside. And if you wanted to see what is happening at the membrane, and if you shine light directly through, can you shine light directly through the medium from the top? Not that. You see, it's penetrating through, right? So it's giving all information in the back of the cell, information inside everything, it's all mixed up. It's too much. You only wanted to see what is going on at the membrane, and therefore, what we do in the microscopy, we change this critical angle until we only light up tiny bit, very small amount at that membrane. That's all what we want, which actually generate semi-super resolution images. Can you go to the bottom one? Right. So we are talking about that the tiny light appearing right there. This small, really small light amount appearing right, just a glittering kind of thing. That's what generates the super resolution. So this 200-year-old concept is developed into a super resolution imaging. I'll show you one of those images. Can you turn the light on? So this is how you see everything, you know, inside out, the molecules in the cell. But when you look at what is only on the membrane, you can see more fine, refined structures. Make it more understandable what those individual molecules do. So the next thing we wanted to show you a little bit about this process called bioluminescence and fluorescence. And you know, there are a lot of organisms. If you go to YouTube, you can see there are a lot of organisms that generate their own light, and then use a process called fluorescence to convert that light into different colors. So in 2008, Shimomura, Shelby, and Rogers Chan won the Nobel Prize for identifying green fluorescence protein and developing it into a biologically useful molecule. So the Rogers Chan actually died a couple of months ago. So first time when I saw him, he was very well running around. Then I saw him on a wheelchair, and then recently I heard he was passed away. But he was a brilliant scientist. So Shimomura used to pull out jellyfish he was from Japan, from the sea, and isolate these green fluorescent protein. And Rogers Chan actually further modified green fluorescence protein to a usable product. So the green fluorescence protein structure is like that. It's the protein. There are a lot of amino acids around the structure which help these proteins to absorb light and does something wonderful. So this is a green fluorescence protein expressing in neurons. We modify this green fluorescence protein and then make neurons to make the green fluorescence protein. So you can see how neurons look like. And so we can do as similar to before. You can actually visualize these neurons cultured in tissue cultured dishes in 4D actually. Length, width, and height over time, that's the fourth dimension is the time. And you can understand this. So we are doing a process, we are just killing the fluorescence in one of these cells using high power laser, and then we are monitoring these fluorescence molecules stacked with the proteins moving from this peripheral area of the neurons into the body of the neuron. So you can understand a lot of biological processes using this approach. So the fluorescence is, I think this is probably one of the take-home messages we would like to deliver to you, the concept of fluorescence. This will be useful in, in the college, probably in the, beyond. It's a very interesting idea where, so you have a molecule, and,
as Professor Rajesh was describing, you have absorption events where light is absorbed, as Kasun showed with that system there. And this picture actually shows out that we do have energy levels. Things are at lowest energy level, which is a ground state. And we have things at excited states. When you're actually causing those absorption events to happen, what you're doing is taking something from a ground state to an excited state, whether it be 1 or 2, doesn't matter right now. We're going up to an excited state. So when you absorb light, you absorb it. But light, we have conservation of energy. Energy just doesn't magically disappear. Things don't like to be in the excited state. So they gotta want to get back to the ground state. So they gotta give this energy off somehow. They can do it by just giving off heat. We call that. Yeah. Yeah, they can give it off by heat, or they can actually just re-emit that light that they absorb. And since we have things in an excited state and ground state, we also have molecules that can move around and vibrate. And we take those into account a little bit. So these are those little vibration states built into each of these ground states and the excited states. So when we absorb up, we have something that maybe goes up to here. And what's going to happen is it maybe decays down a little bit. Actually, better look at this one state here. We absorb the light up, and it gets to this state, and it decays down, gives off a little bit of heat, and then it ends there. And it can continue to give off heat to go all the way back down, or it could give off a ray of light, and this is what happens. So if you look at this, this is an energy level diagram here. This energy right there is a lot bigger than this energy there. So what you see a lot of times with compounds that emit light, the color of light that you put in is different than the color of light that you emit out. So actually it's right here. This is actually an absorption spectrum. The light maximum is right here, but a little bit lower in energy now is actually where the light is emitted.
So, in the visible spectrum, you have blue to red. So blue has the highest energy because the wavelength is shorter, if you just read. And red has the lower energy. So what's happening here is molecule will get excited and lose a little bit of energy, not much. But when you lose energy, and when it comes back to the ground state, it doesn't have the same energy to absorb. So when they emit, it is emitting a different color. So it absorbs one color, and it's emitting a different color. So this process we call Stokes shift. You absorb right here, and lose a little bit of energy, and so it cannot really emit the same amount of same color that it absorbed, and therefore it's emitting different color. So you absorb blue, and you emit green. So this process is called fluorescence, which actually revolutionized the biology forever. Let's see. Okay. Yeah. So here we have the green fluorescence protein that isolated from bacteria. And what we're going to do is we're going to shine a blue light, and you will see. Okay. So if you shine the blue light, so you can see the. So if Kasun were to shine that light right here on the paper, you see it's blue light, right? It's blue light. And if you shine on this protein, it absorbs that blue light and emit a different color, which is we call fluorescence. So high energy absorption, low energy emission, the color change. You guys get the concept, the idea? Okay, all right, cool. So with that, I think we have a few more minutes. I wanted to really get into this optical control of cell behavior. So, you know, the photoreceptors in your eye is like that. They are big proteins, and inside this protein you have a molecule called retina, as I told you earlier, and it is linked to this protein. And when you shine light, or when you look at something with the brightness, this conformation we call cis. It's like a, it's not a chair, right? The board conformation. This is the board conformation, right? Cis. And so this is called a chair conformation. So it goes from cis to trans, and this changed the molecule, this big molecule, activating a lot of things, controlling heart rate or vision or that sort of lot of things. But in this case, actually controlling the vision. So what we did was, we pull out these molecules, light-sensitive molecule from the eye, and then we engineered this molecule so they can talk to the cells not only in the eye, but in the heart, but in the immune system, or in the neuronal system. So we will have these cells, this is a macrophage, produce these proteins on their membrane. And what we wanted to do is, we wanted to activate this molecule to only one part of the cell. You can imagine this cell is about 10 microns. So we have instrumentation in the University of Toledo, so we can actually activate very small region of the cell right there. And then, this is very similar to a phenomenon called cell migration. You know, in a body, when a neutrophil or a macrophage sends a chemical secreted by a tumor cell, it will crawl towards that tumor cell or a bacteria, and it will do this process called phagocytosis, eat them up. So how they do is, they will sense a chemical gradient, low concentration here, high concentration at the target, so they will migrate along the chemical gradient and then find out the target. So here what we wanted to do is, without using a chemical gradient, we wanted to see whether we can control cell migration using light, the light-sensitive photoreceptors from your eye. So there, what you're looking at is two macrophages. You know, macrophages are very abundant in your lungs, eating up all the bacteria and viruses company. So we have attached a red fluorescence tag to these macrophages, and you will see a red box appearing here. What I wanted to do is, I wanted to activate the light-sensitive receptors only one part of the cell, kind of mimicking a macrophage sensing a chemical secreted by a tumor cell kind of situation. So you will see a red box, actually blue light, to activate photoreceptors in this cell, only one part. So let's see. That's the light. You see that? Isn't it cool?
Yeah.
So these are neutrophils. So we are imaging one of the signaling lipids here, which actually control the cell migration process. And we are here imaging a opposite molecule that controls this molecule. And this is an overlay image. So you will see a, I think, blue box, I guess, which is our blue light. That's the light. You see accumulation of this molecule at the leading part of the cell. The cell is kind of diving down. And you turn the light off, the activity goes away. So we are actually trying to see whether we can target the macrophages and these immune cells to tumors or cancers using optical guidance, so that way they can actually fight the tumor growth, that sort of things. So how are we doing at the time?
We have about 5 minutes.
Okay, cool, good. So I showed you two images at the beginning, very beginning, where the flowering plant and a plant moving towards light, right? So in the flowering plant, we talk about this cry domain, cryptochrome domain. So this process called optical dimerization. Simply what happens is, so this is the binding partner of the cry. when you activate cry, when you shine cry, it binds to this molecule. So what you can do is, you can attach a protein that will target this one to any organelle of the cell. Could be the plasma membrane, could be the nuclear membrane, could be the endoplasmic reticulum, whatever. And then you can attach a protein, any protein you like here. It could be a kinase, it could be a phosphatase, it could be anything. And what happens is when you shine light, you can actually move the molecule of interest to any subcellular subcellular location. So this way, in the cell, the bulk volume, your protein of interest will not be active, but when you move it to the target, you can actually drive or control a biological reaction. So for instance, in this case, what we want to do is, we want to activate, we want to actually inhibit a protein, a bunch of protein that is preventing cell migration. So we're going to shine light in one part of the cell. We're going to inhibit a group of protein that are working against cell migration. So, sorry, actually, I'm going to pass this slide. So here what I'm going to do is, now, if you are activating proteins that are working against cell migration, so cells should actually move away from the light, right? So that's what we are trying to see whether we can control. Now you're shining light there. Now it's actually cell is moving. You're just pushing the cell away because you are inhibiting cell migration right there. The cell is actually moving away from the light. So this is a new imaging technique called differential interference contrast microscopy. It's a way to generate nice white light images, so you can actually see the structural changes of the cell. So here is your light. You can see this membrane ruffling generating in the front of the cell. Now cell is crawling towards, actually away from the light, right? So we generated another similar system, this one, using a different set of proteins. This one also working against cell migration. So one can say this is it's a cool technique. What are you going to do? So one thing you can understand, these cells are, this is a 10 micrometer mark, right? And they are very small. You can activate signaling in a really small region, and you can understand the complex behavior or control complex behaviors like cell migration, or actually in this case neuronal growth. So this is an early stage neuron cell, and so this is my blue light right there. We can actually control neuronal growth. You see the growth of a neurite towards light, and you can turn the light off. So this neurite is now consolidating, this structure is consolidating into a neurite. And this is actually, we are thinking whether we can use this technology to kind of repair when you have neuronal damages, guide. I mean, you have the, in the spinal cord, you have the peripheral nervous system. And the spinal cord injuries kind of completely decapacitate people. The problem with the spinal cord is spinal cord neurons grow, but they don't have the directionality. They don't know how to find the right target. They just grow in random directions. So we were thinking whether this technology can be developed so that way one day we would actually guide the damaged neuronal processes towards the right target and then make them reconnect. So these are some of the work we do, and that's pretty much it. And thank you very much. If you guys have any questions, I'm happy to answer.
How long, oh, there we go. How long do you think it'll be between the basic research and the technology being developed?
We are actually collaborating with people that animal models, and from that point, I mean, it could take years. Yes, it will take years, but there's,
Okay, guys, give them a